So we start with the pre-mRNA, which already has the 5 cap on it that creates stability. The snRNPs contain snRNA, which binds to the intron-exon junction by base pairing to certain nucleotides. Other proteins also join to create a spliceosome. And so the snRNPs act as ribozymes, which cleave the intron out from the pre-mRNA, and this intron is degraded. The remaining exons are spliced together by the other proteins, and the addition of a poly-A tail creates more stability, and it's necessary for the mature mRNA to exit through the nucleoli pores.
No, not nuclear pore.
Nuclear pores. Activator proteins bind to the distal control elements of the DNA, and these distal control elements are grouped in the enhancer. There are 3 activation sites, and each of them are called a distal control element. DNA bending protein binds to the DNA and causes it to bend over so that the activators are closer to the promoter. Mediator proteins and the general transcription factors and RNA polymerase II are all nearby at this point. The mediator proteins bind to the activators, and in turn the general transcription factors bind to the mediator proteins, and the RNA polymerase binds to the general transcription factors. This process ensures that it's correctly positioned over the promoter. When this happens, it's called an active transcription initiation complex, and it causes RNA synthesis to be amplified, whereas before it had just been acting at a base level. The diagram represents regulated synthesis of repressible enzymes in the TRP operon of E. coli. Usually TRP operon is on, which means that the repressor is usually inactive. However, in the lac operon, its default is off because the repressor is active usually. So when the repressor is inactive in the TRP operon, it is unable to bind to the operator, which allows RNA polymerase to bind and continue on and for RNA transcription to occur. When RNA transcription occurs, these polypeptide subunits are created, and these code for enzymes that eventually make tryptophan. However, when tryptophan is accumulated in excess, it acts as a co-repressor, binding to the repressor protein, activating it and changing its shape. So it's now able to bind to the operator and prevent RNA polymerase from making RNA, so no transcription occurs and none of these polypeptide subunits are created. TRPR, which is located upstream from the other genes, is the gene that codes for the repressor protein in the TRP operon. This diagram represents regulated synthesis of inducible enzymes of the lac operon in E. coli. Usually the lac operon is off, which means that the repressor is active while lactose is absent, so it's able to bind to the operator, preventing RNA polymerase from going down the length of the DNA and so it does not create mRNA. However, when lactose is present, the repressor is inactivated by allolactose, which changes its shape and so inactivates the repressor, meaning that it's not able to bind to the operator, leaving the pathway open for RNA polymerase to pass through and transcribe these genes to mRNA. LATZ codes for beta-galactosidase, which is responsible for converting lactose into galactose and glucose. LAC-Y codes for permease, which is a surface protein that can allow lactose into the cell. And LAC-A codes for transacetylase, whose purpose is unclear. Also, LAC-I, which is a gene located upstream from the rest of the genes of the lac operon, codes for the repressor protein. So this is a representation of the lac operon, which demonstrates positive control when lactose is present but glucose is scarce. The levels of cyclic AMP are high, so it binds to the inactive CAP, causing the CAP to activate. Active CAP binds to the CAP binding site on the promoter, and CAMP also has to be present, which increases RNA polymerase's affinity to bind to the promoter as well. But once it's bound, large levels of mRNA for the enzymes coded for in the lac operon are produced, so it's like amplification. However, if lactose is present and glucose is also present, the levels of cyclic AMP decrease, and so there's not enough to bind to the inactive CAP, which remains inactive. So RNA polymerase's affinity to bind to the LAC operon also decreases, which means that only base levels of mRNA are produced.